Hi, Bio 111A students. It's Doc Martin here, and I thought I would tell you a little bit about some of the stuff involving things in lab that have been taking place. And what I wanted to say about that is I want to talk a little bit about the business of evaluating your soil isolates for whether or not they have antimicrobial activity. And then additionally, what I would like to do is tell you about something a little odd that came up that will help you. All right? So the first thing that I want to do is to remind you of the kinds of things that we're looking at. Now, again, there's a video that I put together that you should look at. It's in the email that this is attached to that explains to you a little bit about how to look for those zones of clearing. And that's a wonderful thing. But when it comes to Staphylococcus aureus as one of the background organisms that we were checking out, to see whether or not there was any of your isolates had antimicrobial activity against it, we had something odd happen, and I wanted to show you what I meant. And if you are currently looking at this video, at this particular item, what you're going to see is I labeled it properly. What you would normally see for all of your plates is a situation where you have a kind of a cloudy background of the bacteria that you spread all over the surface of the plate. Now, those other colonies all over the plate are some kind of contaminant. My guess is when I subcultured this to go from day 1 to day 2, something snuck in because everybody in the Tuesday section is seeing this. It may be possible to see antimicrobial effects from your soil isolates where you are seeing that cloudiness that you see in the diagram. But the colonies that you're seeing, not A through D, but those colonies in general, as labeled in this image, should show you that you shouldn't be paying attention to those things. I hope that helps a little bit with that. In other news, I wanted to talk to you a little bit about what I would like to see when it comes to your water bottle buddy report. And keep in mind, you'll be doing this with your successful soil isolates as well. So the first thing that I want to show you, and I have a lot of things open, you've got to forgive me on that, is the final version is something like this. Now, if you're looking at this particular image that I'm sharing, you can see the idea of a water bottle buddy. And let me pull that up a little bit so you can look at it. I'm in the habit of suggesting that students not attach their names to this, just because sometimes I like to share these one pages and some students are uncomfortable with it. But it's certainly important to give the date. You'll notice there's a statement here about the water bottle in question, how old it was, how it was washed, and how often, and what they found. There's a photograph that you took of your successful isolate. This isn't a great photograph, but you can see it there. Next, there's the hierarchy view, and please don't worry, I'm going to be explaining all of those things. And then finally, the assignment detail. Now, this is a photograph that you took. This is a trimmed screenshot, and so is this. Down here is a description of that. And what happens is that they go on to show an example. They took this screenshot of a journal article having to do with that organism. Now, as I've said to several of you, everyone is kind of fixated on the idea of something in your water bottle being a pathogen, and that's simply not true. But they can be relatives of them. Methylobacter, for example, is really common in the environment. So what I ask you to do is see if you can find some kind of publication that suggests that the organism that you found in your water bottle is in fact associated with water. And for example, with Pseudomonas, lots of examples of that kind of thing. I hope that helps a little bit. As you look at this though, you say, Doc, I'm not so sure I know how to put that together, though this is attached to the email that you're receiving. So what can I do to make it more clear to you? Well, I am including this. And if you look carefully, what I have here in this particular share is I have a nice illustration of what I would like to see. And here's an illustration of it. Do you see what I mean? the title, and your initials if you don't want to use your name, and I think that's fine. Your lab section and your date, history of your water bottle and such. Do you see photograph of your water bottle buddy as a single colonies, I should say, hopefully not one. Hierarchy view, assignment detail, a little bit of a discussion, and a screenshot of the title and abstract with some relative. And this is also in the email, so you have access to that as well. So you might say, how is that important? Well, what I've decided to do with that is assign some points. And as you can see, and again, this is part of your email, you get the basic rubric of what I'm looking for and how many points are available. All right? So hopefully that'll be a good one. Now, like I said, you have an example that's already completed from another class of mine to give you some guidance. You have the formatting and you have the rubric for that. But I also found that some students were a little confused or asking me questions on how best to analyze what they had. And I wanted to take a moment with an example of that. So remember, you have two isolates with your number and your letter after them. And the first thing that I asked you to do is to look at those chromatograms. And so here's an example from your class. And what you're looking at here is a particular chromatogram. Now you'll notice that starting about here, the peaks are very nice and distinct throughout, and they go for a long way. This is a really, really good chromatogram. It starts to fall apart toward the end. Remember, the beginning and the very end aren't as reliable as the rest. But remember that you must look at the chromatogram and hopefully see that you have a good one, because that'll help inform what you're doing. Keep in mind that when you do that, if you think about it, what you have mostly is just material like this, where you have that text file. And that text file, you can't tell if the data is good or bad. That's really the value of that. So let's go ahead, as I showed you in lab, and let's copy. Oh, I had a student ask me, what if I had both of my chromatograms that are great? Well, that's wonderful for you. And that might be helpful to people in class who had situations that didn't work out. In any event, I cut and I pasted that together, the nucleotide sequence. It'd be nice if I could speak clearly. Thank you for your patience. And then what I do is I go through the first of two different programs to look at it. And the first thing that I do is I pop over again to what we discussed in lab, and you have the website for in a previous email, RDP classifier. And when you paste in that particular sequence that I just copied, and you pop that in, and it thinks, and it thinks, and then it thinks some more. And would you look at that? It's just beautiful. Here's the hierarchy view. This is that one part in the water bottle buddy that I mentioned. And it shows you how things work from all the way from the domain bacteria down to all the way to the genus Xanthinobacterium. When you click here, show assignment detail, that shows you how much you can trust that data. And as you can see, it's all the way down to the genus level of Xanthinobacterium. So this is really, really good data. And so this is very trustworthy. I know that this water bottle buddy for this student is Xanthinobacterium, which is great. I also told you, and this is the assignment detail that you also need for your one pager. Now, then when you go to the other program, and you might say, what's the point in doing that? Well, you want to make sure that they both kind of agree, especially if you get data that's a little bit challenging. For example, you can trust anything over 80%, but what if this was 78%? You're going, if it's the best I've got. But if you go to BLAST, you can check it. And you go ahead and you put that nucleotide sequence in there. You blast away. And as I told you before, sometimes this takes a minute or two to finish. And that's what's taking place right now. It's thinking about it. And then it thinks some more. And then it thinks still more. I'm trying to make things, everything as clear as possible with you. It's really important to me. Now, while it's thinking, I want to remind you that what I wanted from folks on Monday is a chart of your soil isolates and the effects they have on the 7 different bacteria that you have. And that's the business I talked about, with minus meaning no zone of inhibition, plus minus sort of a zone, plus is a zone, plus is a huge zone. So you're going to have this great chart that we can talk about in lab. That'll be exciting. All right. And that's for the soil isolates. Now we're working back to water bottle buddies to finish what we're talking about here. And it's still thinking. I feel funny saying to you, thank you for the patience of this program, with this program. But here we are. And the wonderful news that you can see is that you get lots and lots of Xanthinobacterium. So that tells you that your blast data and your RDP classifier data agree, which is perfect. So let's do something. Let's just, I don't know which one to pick, but you'll notice the max score is great and the percentage identity is great. And I told you before that when you click on one, and today let's do this one right here. And when I do this, I have to get over to the toggle. There we go. It just shows how similar things are. But I really want you to go over to GenBank that I just clicked. And we'll see if this helps, because it might not. Like I said, I'm doing this just like the rest of you. And this is, in fact, from a paper. All right, this is great. If you look here, it's from a journal. And there's even what's called a PubMed description of it. And when you click on that, it thinks, and it'll take you to this. Now check it out. And look at this. Do you see where the isolates were isolated from? That's too many isolates. The isolates were first identified from environmental water ponds. That is to say that oligotrophic means low nutrients. So this means kind of like your water bottles. This is great. That means it's not unlikely that Xanthinobacterium is in water bottles. And so what you would do is you would take this particular situation that has the abstract and the title and the journal, and you would make yourself a nice screenshot of that. And that would be the last thing that you would add to your water bottle buddy write-up. Does that make sense to everybody? I hope that it does. So again, what I did is I took you through the process of putting together your water bottle buddy candidate in a way that for that one-page thing that I would like to see. Again, attached to the email that this link appears in, you're going to be seeing examples of all of these things to help you. Now, for both the chart of your soil isolates and the effect they have on the 7 different bacteria, and then the water bottle, I would like both of those on Monday. And that'll give you an opportunity to contact me if you have any problems understanding what to do. For example, you might have a situation where you have 2 great isolates. You get to pick one. That's not a problem. What if both of yours are not good by the chromatogram and by RDP classifier? You're going to find that there's a folder on the shared drive that's going to read additional water bottle buddies. And what it's going to include is some material with a photograph of some colonies, with a chromatogram, and with the nucleotide sequence, the text file that I told you about before. So the point is, even if nothing worked, I've got you covered. All right? Well, I'll be seeing you in class pretty early tomorrow. So I hope you have a wonderful evening, and I will see you later on. Best wishes to all of you. And thanks again for taking this class and doing this very cool lab exercise. Bye now.